Dear students, today we are going to discuss about the gene expression during the development of photosynthetic machinery. In the previous lectures, we have seen that the chlorophyll biosynthesis that takes place from the amino levulinic acid to the synthesis of chlorophyll A via 14 different types of enzymes and steps that are involved over there. Then the degradation of the chlorophyll molecule, that is the reversal of all the processes that are taking place during the chlorophyll biosynthesis. We have followed the Bailey pathway of the chlorophyll biosynthesis and the phosphorbite pathway for the degradation of the chlorophyll molecule. We have seen that the green chlorophyll molecule that is degraded to non-fluorescent catabolites, and then these non-fluorescent catabolites, they become soluble in water. Otherwise, chlorophyll molecule is insoluble in water or other soluble solvents. Only methanol, acetone, benzene, or petroleum ether. These are the solvents for making the chlorophyll molecule soluble. Now, all the development of the chlorophyll molecule are the development of the photosynthetic machinery that takes place in the presence of light. So when this is the development in the living systems, this cannot take place without the help of enzymes. And that means when we say that it cannot take place without the help of enzymes, that means there is quite a lot of gene expression that is going on for the development of the photosynthetic machinery, anchoring into the photosynthetic membranes. So the regulation of the nuclear and the chloroplastic gene, that is important because the RuBPCase, this is which is ribulose 1,5-bisphosphate carboxylase oxygenase. It is a protein which is important for the in the dark reactions of the photosynthesis. This protein has two subunits. One is the large subunit and other is the small subunit. Large subunit of RuBPCase, that takes place, the development of this large subunit of RuBPCase, that takes place in the chloroplast, while because the gene for its synthesis that are located in the genome of the chloroplast. Whereas the small subunit of RuBPCase genes, they are located in the genome of the which is found in the nucleus, and their messenger RNA synthesis that takes place in the nucleus, which is then transported to the cytoplasm. So this requires the regulation. So the regulation of nuclear and chloroplastic gene expression, that is principally studied during development, particularly when we are seeing the greening and degreening. Greening means the conversion of etioplasts to the chloroplast, and degreening, that means the conversion of the chloroplast either to chromoplast or to etioplasts. So in the instant case, or in the synthesis of the chlorophyll of this chloroplastic gene expression, the light dark cycles, they are important. So this is a great approach in which the leaves, they are first exposed to light environment, then dark environment, and then light environment. So during all this light dark cycle, or the light dark transition, there is the upregulation and downregulation of different genes. and this can take place in response to certain stress factors as well, such as high temperature, low temperature, radiation stress, etc. So all these the light dark cycle or the heat cold or radiation stresses, they have shown that the multiple levels of genes are regulated for most of these genes which are involved in the development of the photosynthetic machinery. Yes, this is difficult to generalize, but some trend of the expression of these genes. Now, the translational regulation is generally global. That is, most of the genes, they show increase or decrease together. And most of the plant species, they show the same trend. To exemplify it, overall expression increases during the greening, that means upon exposure to light, and this decreases during the conversion of the chloroplast to chromoplast or the chloroplast to etioplast, when this is the case of the developmental queue in the development of the chloroplast to chromoplast, or in the development of the etioplast when the leaves, they are exposed to darkness. There are the examples of gene expression translational regulation. PSBD, PSBC promoter switches in response to light. Now the regulation of RNA splicing and stability. Splicing, as we have studied previously in the lecture on ribosome, that the RNA causes its own cutting and rejoining. So the splicing of some photosynthetic genes, that is the intron group 2, is inefficient in maize roots, amyloplasts. Splicing of psbA intron, that is the group 1 intron, in Chlamydomonas, which is an alga, is strongly promoted by light. Stability of some plastid messenger RNA increases during greening and are between light and dark cycle, as has been studied in clay. The chloroplastic messenger RNA are relatively long-lived. Their half-life is 0.5 to 8 hours or even more than that. So the translation is regulated in the chloroplast by global changes in the rate, that is the light dark cycle, and this is high in daytime because of the availability of light and low at night time because of the absence of light. Preferential translation of the specific messenger RNA under certain conditions, that is very high during light intensity, increases the psbA translation and decreases the ribulose biphosphate carboxylase large subunit trans. So here before you is the model in which you can see that the both the synthesis of large subunit of ribisco and small subunit of ribisco, that is taking place separately in two compartments. This is the cell, and this is the nucleus, and this is chloroplast. So whenever there is a need for the synthesis of a functional ribisco, then a message is sent from the chloroplast to the nucleus for the synthesis of small subunit of ribisco. Here what happens is that the gene for the small subunit of ribisco, that is expressed, and messenger RNA, that is synthesized. Likewise, at about the same time, the gene for the large subunit of ribisco, which is present in the chloroplast, that is expressed, and large subunit messenger RNA is synthesized. Now in the nucleus, the small subunit of ribisco RNA, that goes into the cytoplasm, where the small subunit of ribisco protein, that is synthesized. Now this protein, after its initial processing, leads towards or heads towards the chloroplast, where the large subunit of ribisco protein is present. Both of these combine to give rise to the functional rubisco. Now this all is taking place in the presence of light. So here we can, from this model we can demonstrate that the light activates the translation of psbA or the photosystem to protein D1 messenger RNA. So here an assay is used which is called as the gel shift assay. Here as you can see from this gel that there is three lanes are marked, 1, 2 and 3. The lane 1 is the control where no protein extract has been added. So this is just the water added or just the reactants that are used for the extraction of protein, they are added in this lane. Lane 2 is the extract from the light grown cells. This is lane 2, and this is the extract from the light grown cell. And lane 3 is the extract from the dark grown cell. So this is lane 3 and dark grown cell extract for the protein of the psbA or the D1 protein, they have been added in all these three gels. When these gels were run, it was and visualized using appropriate equipment. So it was noted that in the first lane where no extract was loaded, there is nothing present in that. In the second lane where the extract from the light grown leaves were added, we can see a strong protein messenger RNA complex that has been visible. Very strong expression has been there. And in the lane 3 where we have added the extract from the dark grown cells, hardly any expression of this protein messenger RNA complex is shown. So this indicated that light activated the translation of the psbA photosystem to protein D1 messenger RNA, and which was functional as well. And in the darkness, this free messenger RNA was found, which is rapidly prone to the. Now proteins in complex that bind to the 5 and untranslated region of the psbA. Here PABP, this is similar to poly A binding protein that binds to AU rich region in the 5 and untranslated region that activates the translation. PDI, this is a protein disulfide isomerase that reduces the disulfide bond on certain proteins and activates the PABP to bind to RNA. Third is the kinase that responds to ADP level. At high ADP, deactivates PDI by phosphorylation. Now we'll keep in mind all these three terminology. We will see apply this terminology over here in this model, which is the model for activation of psbA translation by light via photosynthesis. So here we can see that this is the photosynthetic membrane, that is the thylakoid membrane, that shows the PS2, which is in the presence of light. There is the splitting of water. The electrons are excited, they go uphill, and then these electrons they move to the PS1, photosystem 1, from photosystem 2 to photosystem 1. Now again, upon the availability of light, these electrons they enter into the electron transport chain. Now if the light is available, then everything that will go normally, and the translation that will be on. That means there will be the formation of messenger RNA for the synthesis of protein for the photosystem proteins. So here we can see that this is a PDI, this is a PABP, and the kinase that is responsible for the synthesis of ATP. So the kinase that will lead to the synthesis of ATP in the presence of light. The presence of light is a reducing environment, and the absence of light is an oxidizing environment. So when I say that the kinase is functional in the reducing environment or in the presence of light, that means there will be low level of ADP. That means the ATP that will be continuously used for the biosynthesis of ATP during the light reactions of photosynthesis. Whereas when we talk of the oxidizing environment or the absence of light, the ADP level will accumulate, that will be higher, and the kinase that will not be functional in the oxidizing environment. So the kinase activity will be inhibited, and eventually there will be the translation that will be shut down. So in the reducing environment, there is that is the light environment, the translation will be on, and in the darkness or oxidizing environment, the translation of the protein that will be. Now the regulation of protein stability in the chloroplast, this is triggered by again the binding of the chlorophyll molecule to the photosystem proteins. Now one thing that we must keep in mind is that the protein, the photosystem protein, they are the apoprotein. The chlorophyll molecule that acts as the cofactor. So the binding of this chlorophyll or the cofactor to the photosystem protein, that will make the hollow protein of the photosystem. So this hollow protein is the functional protein, and what happens is that the chlorophyll molecule, that is the harvester pigment, so it is exposed to the outside environment for harvesting light. So this harvesting of light that makes the photosystem functional. The chlorophyll molecules, they are lying like the antenna, and they will harvest light, pour onto the photosystem too, and so the assembly of the chromophore or the cofactor, which is the chlorophyll, to the photosystem protein, that is taking place in the presence of light. So the binding of the cofactor, that is important, and the assembly with other subunits in multi-subunit enzyme complex, that is again importance. So here in this model we can see the stabilization of the nascent chlorophyll binding proteins of PS1 and PS2 with the chlorophyll. So here this is a simple model that shows that in the presence of light, the chlorophyll synthesis will take place, as we have seen in the previous lectures. And if the light is not available, then there will be no chlorophyll synthesis. So if the chlorophyll synthesis is there and the protein, photosystem proteins, they have been synthesized, then in the presence of light, the chlorophyll will bind to the protein, and a stable photosystem will be formed that will be present in the thylakoid membrane. And when there will be no chlorophyll synthesis due to the absence of light, the proteins, if synthesized, they will remain unstable and will be degraded. So light is important for stabilization of the nascent chlorophyll binding proteins of PS1 and PS2 with the chlorophyll, as we have understood from this. Then photoinhibition. the photoinhibition takes place at the time when there is the presence of light over and above the required amount of light. This excess of light that will inhibit the photosynthesis, and this has a great impact on the plant performance because excess of the light that will damage the photosystem. So how we can know that the photoinhibition has taken place? This is assessed in terms of the rate of photosynthesis, and that is measured in terms of the oxygen evolution, because in the presence of light, the photolysis of water, or the splitting of the water molecule that is taking place, the oxygen evolving during photosynthesis is originally from the splitting of water. So the molecular oxygen that will be evolved. So when we go from low light intensity to high light intensity, the rate of photosynthesis that continuously increases, and at a point this rate is saturated. So this light, when this is there, the rate of photosynthesis that is saturated, and if excess of light is further available, then this saturated rate of photosynthesis that will begin to decline due to the photoinhibition, and this will be called as the photoinhibition range, where a decrease in the photosynthesis or a decrease in the oxygen evolution that will take place. So this all will lead to the this will indicate the damage to the photosystem too, and then to the as well. The psbA encodes 20 to 32 to 35 kDa D1 polypeptide of PS2. So these are D1 and D2. These are the proteins that are the photosystem 2 complex protein. Of these two proteins, D1 is more important because D1 turns over rapidly, because it becomes damaged by the excess of light. So its replacement is continuous, but if the light is in photoinhibitory range, then D1 protein of the PS2 is more fastly damaged as compared to its synthesis. So when this is more damaged than its synthesis, that means there is a continuous damage to the photosystem, because the replacement rate that is much lower as compared to the damage rate. So at most of the lower light intensities, the degradation, synthesis and replacement of D1 keeps up with the damage rate. So at low light intensity, this is a normal process, that the D1 that continues to degrade, and its replacement is almost at same rate. Then nuclear control of the chloroplastic gene expression. Genetic studies have revealed considerable potential for the nuclear control of the specific chloroplastic genes. Here the mutants they have been used to study the nuclear control of the chloroplastic gene expression. Many of the Mendelian or the nuclear mutants that are defective in the plastid development or function do not express at a specific chloroplast encoded protein. So such type of mutants, they have been reported from Chlamydomonas, which is an alga, Arabidopsis. This is a model plant for the study of many of the biological processes in plants, and maize. This is another plant, the genome of which has cloned. So as a result of the use of these mutants in Chlamydomonas, it has been seen that a number of chloroplastic genes are expressed, and they affect different steps during the protein synthesis, and the loci, number of loci that are on which these genes are present. So psbA, this affects the translation, and it's present at 2 loci on the chromosome. psbB, this is responsible for RNA stability. 1 locus is present. psbC, this is again responsible for translation. 1 locus present. psbD affects the translation. 1 locus present. psbD, RNA stability. 1 locus present. RBCL, ribulose bisphosphase carboxylate, large subunit. This is responsible for RNA stability. 1 locus present. PSAA, this is trans-splicing. 14 loci are present. ATPA, RNA stability, 1 locus. ATPB, translation, 1 locus. And RBCL, transcription, 1. So the nuclear genes that encode the proteins for photosynthesis are expressed poorly if plastid do not develop. So for the expression of these nuclear genes, the plastid development is again very important, or are damaged by photooxidation. So this suggests that a signal from the plastid to the nucleus that is taking place, because as I told you earlier that the gene for the expression of the small subunit of rubisco that is present in the nucleus, while the gene for the expression of the large subunit of rubisco that is present in the chloroplast. So the message that will come from the chloroplast towards the nucleus for the expression of the relevant genes. This signal may be small molecule or intermediate in the chlorophyll biosynthesis. So the chlorophyll biosynthesis is very important step in the whole of the maintenance of the photosynthetic machinery. So dear student, that concludes your midterm course, and this is my email address. If you have any questions or queries, you can address your questions or queries by email on this address. I will urge the class representatives to spread this animation to all the students so that they can benefit from it. Thank you very much.